Hello everyone, welcome back to our YouTube channel. Today, I am going to talk about Chapter 6, Plant Nutrition. This is for IGCSE CIE Biology 0610 students. I am Dr. Mary, and let's get started. First of all, what is photosynthesis? Photosynthesis is the process by which plants synthesize the carbohydrates from raw materials, such as carbon dioxide and water, by using the energy from light. So here you can see two equations. The first one is water equation and second one is balanced chemical equation. When the exam question asks you to write the water equation, you must make sure that the substrates and the products are totally correct. This is also the same for balanced chemical equation. So you have to write the symbol of the molecules correctly and also the coefficient number 6 here. What is chlorophyll and what does it do? Chlorophyll is a green pigment that is found in chloroplasts of plant cells. The chlorophyll reflects the green light, the green part of the light. Which means that the chlorophyll does not absorb the green part of the light, but it does absorb the red and blue parts of the light, so that the plants have the characteristic green color. Chlorophyll absorbs the light energy. Its role is to transfer the energy from light into chemical energy that will be stored in the form of carbohydrates, such as glucose and starch. Photosynthesis will not occur in the absence of chlorophyll. For example, the cells in the roots do not have chlorophyll. That's why they cannot do the photosynthesis. How are the products of photosynthesis used? The glucose product from photosynthesis can be converted into starch, which is usually stored in tubers and seeds. The starch does not have the osmotic effect, while the glucose has the osmotic effect. That's why the plant usually stores the food as starch to remove that osmotic effect, the effect of drawing the water into the cell. That glucose can also be converted into cellulose, which is the part of the plant cells, the cell walls, which is needed for the cell walls. Yeah. The glucose can also be converted into sucrose, which is needed for respiration to release energy. That sucrose and another form, fructose, can be used as nectar, can become nectar to attract the insects for pollination. That sucrose can also be used for transport in the plants. Then the plant can also convert the carbohydrates into lipids for energy storage inside the seeds, which will be used for germination. The plants can also convert the carbohydrates into amino acids when they are combined with nitrogen or nitrates, which are absorbed by the roots by active transport. Now let's look at the table of the important mineral ions for the plants. Although there are many other important mineral ions, these two are the most important ones for your exam. The first one, magnesium. Magnesium is used to make chlorophyll for photosynthesis. When it is deficient, the leaf will become less greenish and will become more yellowish. That is why the leaves of the plant will look like this, yellowish discoloration of the leaves. The second one, nitrate. Nitrate is used to make amino acids and so proteins. When they are deficient, it will cause the delayed growth of the plant. Now, let's look at the investigation part of this chapter. Before we move on, you have to understand this concept. When photosynthesis occurs, it uses up the carbon dioxide from the surrounding. There will be less carbon dioxide in the surrounding, such as atmosphere. And also it takes up the water from the surrounding, for example, from soil. Then those two are converted into glucose. That's why the chlorophyll will have more glucose and starch. And also oxygen is produced. That's why more and more oxygen will be released into the surrounding. So these concepts will be used in the later investigation procedures. Now let's look at the de-starching part of the investigation. If the presence of starch is your evidence in the photosynthesis investigation, you must make sure that there is no starch in the leaf at the beginning of the experiment. This is because any starch already present in the leaf will affect the results of the experiment. It can confuse the result of the experiment. That's why we need to make sure that all the starch already present in the leaves before the investigation are used up before the investigation. This can be done by leaving the plant in the darkness for a few days. Now, let's look at how do we test a leaf for starch. A leaf that is about to be tested has to be drawn into the boiling water to kill the cells and to break down cell membranes. Then the leaf will be left for a few minutes, for example, 5 to 10 minutes, inside the test tube that is containing hot ethanol. This removes the chlorophyll, so color changes from iodine can be seen more clearly. Then we dip the leaf in boiling water again to soften it. Then we take out the leaf and spread it out on a white towel to see changes clearly, and we cover the leaf with lots of iodine solution. The parts containing starch, which means the photosynthesis happened, will turn into blue or blue-black color with iodine, while the other parts without starch will remain brown or yellowish with iodine solution. So we can look at the photo here. The green part, which contains lots of chlorophyll, will do the photosynthesis a lot in the presence of light, carbon dioxide, and at a suitable temperature. So there will be lots of starch. Those starch will give the iodine color changes into blue-black. So the white part here does not have any chlorophyll, so it will not photosynthesize, so there will be no starch, and so the iodine in those parts will remain brownish. You have to note a safety precaution here, that you have to make sure the Benson flame is turned off before you start heating the ethanol, because ethanol is flammable. Then we partially cover a leaf of a plant with aluminum foil, just like in this picture, and we'll place that plant in sunlight for a day. After that, we remove the leaf that is partially covered from the plant, and we'll test the leaf for starch by using iodine, just by using the same procedure that I mentioned earlier. This slide is hello. Now let's look at the investigation of the need of light for the photosynthesis. Now let's look at the investigation of the need of light for the photosynthesis. First of all, we need to discharge the plant by placing it in a dark cupboard for 24 hours because we are going to use the presence of starch as an evidence at the end of this investigation. Then we partially cover the leaf of the plant by using aluminum foil, just like in this picture, and we place that plant under the sunlight for a day. After that, we remove the leaf that is partially covered and test it for starch by using iodine. The area that was covered will remain brownish because there was no photosynthesis happened in that area. The exposed part here will do the photosynthesis, so there will be lots of starch. So it gives the blue-black color. Now let's look at the investigation of the need of carbon dioxide by photosynthesis. Since we are going to use the presence of starch as an evidence in this investigation as well, so we need to discharge the plants, the two plants we are going to use in this investigation by placing them in the dark for 24 hours. Then we place the beaker of soda lime in one plant so that it will absorb the carbon dioxide from the surrounding. Then we place a beaker of water in another plant. This plant will become control experiment. The beaker of water will not absorb any carbon dioxide from the surrounding. So the plant with the beaker of soda lime will not get enough carbon dioxide for photosynthesis, and the plant with the beaker of water will get the normal level of carbon dioxide from the surrounding. Then we cover both plants with plastic bags. Then we place both plants in bright light for several hours. After that, we test both plants for starch by using iodine. The leaf from the plant without soda lime will turn into blue-black because it can photosynthesize normally. Now let's look at the investigation of the effect of changing light intensity. How can we change the light intensity? We can do it by moving a lamp from distance to distance, different distances, away from the beaker containing the pond wheat. So the farther the lamp from the pond wheat, the lower the light intensity. So this is the method that we use. And how do we know that the pond wheat is doing the photosynthesis or not? We can observe that by observing the bubble release from that pond wheat. As you can see in this picture, the bubbles are appearing from the cat end of the stem. So the more the bubbles release from the cat stem, the faster the photosynthesis happens. We can assume the conclusion like that. But this is not a very good evidence because the bubbles may contain oxygen or carbon dioxide or any other gases. We cannot make sure that the bubbles contain oxygen, which is produced from the photosynthesis. So there will be more and more follow-up question in your investigation question, like how can we improve this investigation design? We will talk about it more when I record the past paper question videos. Now, how do we investigate the effect of changing temperature? So we can do this by changing the temperature of the water surrounding the pondweed by using water bath and thermometer. So just like the previous example, here we can demonstrate that the pondweed is doing the photosynthesis or not by observing the bubble release from the cat stem of the pondweed. So here is an important note. Whenever you are writing or analyzing the investigation design, you have to keep in mind that all the variables must be constant, except one condition, in order to make sure that those tests are fair. For example, like in this picture, when we investigate about the effect of light intensity, only the light intensity has to be variable, has to be different. The other variables, such as temperature, must be kept constant. But here is a pitfall. Since we use a lamp to change the light intensity, the lamp also gives off the heat. So not only the light intensity, but also the temperature will change in this investigation. So the tests are not fair anymore. To remove that error, to improve this investigation, we have to keep a tank or a beaker of water between the pondweed and lamp so that the water will absorb the heat from the lamp. This is how we improve the design of investigation. Now, let's look at the part of limiting factors. What is a limiting factor? A limiting factor is something present in the environment in such short supply that it limits life processes. So, what are the main limiting factors for photosynthesis? Those are temperature, light intensity, carbon dioxide concentration. Light intensity. Light is one of the requirements for photosynthesis. So we can assume that when there is more light, there will be more photosynthesis. It is partially true. But it can only increase the rate of photosynthesis only until a certain point. Because at that certain point, regardless of how much light we give, since other requirements for photosynthesis are not enough anymore, such as carbon dioxide, the rate of photosynthesis will not increase as much as like before. So you can see here at low intensity, the rate of photosynthesis increases quite steeply. But here at one point, it decreases. The rate of photosynthesis become slower compared to this level. And then it become much, much slower and will not increase anymore at some point. So how does the temperature limit the rate of photosynthesis? As temperature increases, the rate of photosynthesis also increases because the reaction is controlled by enzymes. Here we have to understand the effect of temperature on the enzyme catalyzed reaction. If you are not very familiar with that concept, please watch the video that appears on the right upper corner of this video. So let's continue with this topic. So just like any other enzymes, the enzymes in the photosynthesis will perform much faster until the optimum temperature. At the optimum temperature, the enzyme catalyzed reaction will occur fastest. After that, the heat will break down the structure of the enzyme because they are proteins. The enzymes denature. That's why the rate of reaction after the optimum temperature will become slower and slower, just like in this graph. Now, the last limiting factor, carbon dioxide concentration. Since carbon dioxide is also one of the raw materials required for photosynthesis, this means that the more carbon dioxide that is present, the faster the reaction can occur. So similarly, this trend will continue until some other factor required for photosynthesis prevents the rate from increasing further because it is now in short supply. Now we move on to the leaf structure part of this chapter. This chapter is a bit lengthy, but it is quite important for your exam. So here, the leaf structure. Let's look at the uppermost part of the leaf, which is cuticle. Cuticle is actually not a cell layer. It is simply wax layer. Those cuticle wax is secreted by the upper epidermis cells. So the upper epidermis cells are here. You can see those are quite clear so that they look transparent and they do not block the absorption of light by the cells beneath them. So the cell layer beneath the upper epidermis is palisade mesophyll layer. The palisade cells contain lots of chloroplasts, as you can see here, and the shape is long and columnar. This is the main site of photosynthesis. The layer beneath the palisade mesophyll layer is spongy mesophyll layer. So these spongy cells are quite spherical and rounded, and they are loosely packed. There are lots of air spaces in between those cells. Those air spaces are used for gaseous exchange. So between those mesophyll cells, there are vascular bundles, as you can see here. Those vascular bundles are used for transport in plants. So I will talk about this transport in plants more in that chapter. After the spongy mesophyll layer, there is again another epidermis layer. This is called lower epidermis layer. The lower epidermis layer contains the same transparent cells, but It also contains the guard cells and stomata. So those stomata are used for gaseous exchange, for diffusion. You can see the guard cell more closely here. So these are two guard cells. In the middle, there is an opening called stoma. Stoma is just an opening. It is not a cell. So through that stoma, the diffusion of carbon dioxide into the cell, into the leaf, and the diffusion of oxygen and water vapor out of the leaf occur. So I summarized the description that I mentioned earlier in previous slides in this table. So you can pause the video here and note this description down into your notebook. I'm also underlying the important points here, as you can see here. Now, let's look at the table of adaptation of leaf structures for photosynthesis. This is quite important for your exam. Biology papers usually ask the questions relating to adaptation. For your exam, you have to remember at least 3 adaptations from this table. Now, let's look at it. The first one, large surface area. This is the usual adaptation feature by most parts of the living organisms. This feature is quite an important adaptation to increase the rate of diffusion. Here in the plant, in photosynthesis, to increase the rate of diffusion of carbon dioxide and absorption of light for photosynthesis. And the leaves are thin. This is to shorten the diffusion distance for faster rate of carbon dioxide diffusion. And in palisade cells, there are lots of chloroplasts, and so lots of chlorophyll. This is to absorb lots of light for faster photosynthesis. The epidermis layers are quite thin and transparent, especially the upper epidermis. This is to allow more light to reach palisade cells for faster photosynthesis. The uppermost cuticle layer is thin and made of wax. This is to protect the leaf. At the same time, not to block the sunlight. The spongy cell, the spongy layer. There are lots of air spaces for faster carbon dioxide diffusion. Stomata. They are there to allow carbon dioxide diffusion into the leaf and to allow the diffusion of oxygen out of the leaf. Now let's look at the pathway of carbon dioxide from the atmosphere to the chloroplasts. This occurs by diffusion. So, according to the title here, the pathway will start from the atmosphere. So here, atmosphere, from the atmosphere, carbon dioxide will diffuse through the stoma into the air spaces, which are between the spongy cells, spongy mesophyll cells, and diffuse through that air spaces, and the carbon dioxide will reach the palisade mesophyll layer. There, they will diffuse into the carbon dioxide molecules will diffuse into the palisade mesophyll cells and then into the chloroplasts, where they take part in the photosynthesis. So here is the investigation about the gas exchange in plants. It is also known as the investigation of the effect of light on photosynthesis. Here in this chapter, you will see the similar title, the effect of light intensity on photosynthesis. There are 3 investigation designs in this chapter. I will recall them. Follow me. This is the first one. Here, we use the presence of starch as an evidence that the plant is doing the photosynthesis. This is the second one. Here, we use the release of bubbles, the number of bubbles released from the cat stem of the pondweed as an evidence. Yeah, third one. Here we are going to use the level of carbon dioxide in the surroundings, whether it decreases or remain the same as an evidence here in this investigation. Before we move on to the investigation, design and results, we have to understand this concept first. During the dim light, not only during the dim light, throughout the day, throughout every day, the living organisms, including the plants, have to do respiration to release energy so that they are alive. So the respiration happens all the time, not only in the dark hours, but also in the light hours throughout the day. Since the respiration take up and use the oxygen and release carbon dioxide, those released carbon dioxide will be used by photosynthesis. So during the dim light hours, the rates of respiration and photosynthesis are equal. So there will be no excess carbon dioxide. So no carbon dioxide will be released from the plant, from the leaves. The same, the oxygen released from the photosynthesis will be used up by the respiration. So there will be no oxygen diffusion out of the leaves. So during the dim light hours, there will be no changes in the surrounding oxygen and carbon dioxide levels. During the bright light hours, the photosynthesis happens much faster. So it needs lots of carbon dioxide. So it uses up the carbon dioxide not only from the respiration, but also from the surroundings. So when there is a faster rate of photosynthesis, the level of carbon dioxide in the surrounding, in the atmosphere will decrease. This can be tested by using hydrogen carbonate indicator. On the opposite, during dark hours, when there is no sunlight, when there is no light, there will be no photosynthesis. So the carbon dioxide from the surrounding will not be used up. Not only not used up, the excess carbon dioxide which are being released by respiration will be released into the surrounding. So there will be an increase in the level of carbon dioxide in the atmosphere. So when there is an increase in the level of carbon dioxide in the atmosphere, it means there is no photosynthesis happening. So we will use that evidence in the following investigation. So this is the hydrogen carbonate indicator that I mentioned earlier. It is actually a pH indicator. When it changes to yellow, it means there is an acidic pH. So here in this case, since carbon dioxide is an acidic gas, that increase in carbon dioxide will give a lower pH. So the indicator will change into yellow. When there is less carbon dioxide, the pH will be higher. So the indicator will change into purple. At the atmospheric carbon dioxide level, the normal pH will give a red color. Keeping that in mind, let's look at these test tubes. The number 2 test tube is put inside a cupboard, which means the light is blocked. And the indicator shows yellow color, which means there is an acidic pH. So since there is a pond weed here, we can assume that this acidic pH is due to an increase in carbon dioxide level in that solution. So this means there is no photosynthesis happening inside that pond weed. Let's look at this number 1 test tube. The color of the indicator is now purple, which means there is a higher pH. Since there is loss of light and there is a pond weed inside the test tube, we can assume that that purple color is due to a decrease in carbon dioxide level in that test tube, which means there is lots of photosynthesis happening inside that pond weed. The test tube number 3 here, it contains no pond weed. This is used as a control test for comparison with the other 2 test tubes. We can see the color of the indicator, which reflects the atmospheric pH that in that test tube. So we can compare the color with the other 2 test tubes, and we can make sure that these color changes are actually due to the changes in the carbon dioxide level, or at least pH in that test tubes. So this is the end of this video. I hope this is very helpful for you guys. If you like this video, please like the video and please subscribe our YouTube channel. Thank you so much for watching and see you again in next video. Bye-bye.